frequency.
Video, I'm going to be talking about the MVB loading into the sacs dynamics and some of the machinery involved. So basically this has to do with exosomes, and the aim of the video is to stop or prevent exosomes from forming and releasing in the first place, because once cancer exosomes do that, they can cause a lot of havoc, a lot of issues, even supposedly irreversible issues, but I don't think it's irreversible. I think there could be a way to reverse it, but a good way is to prevent it in the first place, which is the havoc that exosomes cause. The mechanism of action is that ORPs, short for oxysterol-binding protein-related proteins, direct or indirect binding. Oxysterols can bind directly to ORPs, which act as lipid transfer and sensing proteins. So ORPs can shuttle sterols between membranes or present sterols to other machinery. So there's this thing called ubiquitin, and basically oxysterols accumulate in regions of the membrane where proteins have been tagged with ubiquitin. This ubiquitination signals ESCRT complexes to sort these regions into ILVs, or short for intraluminal vesicles. The way to stop this ubiquitination signals, because if they are ubiquitinated enabled, then basically the oxysterols they will be loaded onto the intraluminal vesicles, or those vesicles load the oxysterols onto the MVBs, which is the aim or the target is to stop that from happening, and that is by ubiquitinating the membranes. By doing so, ubiquitin efficiency is a lot lower, since the membranes where the oxysterols are accumulated at are tagged with ubiquitin. So as a result of the lack of ubiquitination, slower ILV formation, less selective cargo sorting. The cargo would be the oxysterols. Okay, there are adhesion molecules, which are integrins and ICAMs. Basically what they do is that their extracellular domains can mechanically stabilize budding ILVs. So oxysterols or ORP activity can change lipid composition and enhance or inhibit integrin clustering, affecting ILV formation, which is the very thing that this video is targeting, to prevent the formation of ILV, because the ILV will aid in the oxysterols loading onto MVBs, and then once that happens, the exosomes are formed and released, which is what I'm trying to stop. Okay, so even if ubiquitin is weakened or disabled, these things called tetraspanins can still act as scaffolds, stabilizing areas of budding vesicles, which are the ILVs. So the tetraspanins, which are CD9, CD63, and CD81, also have to be targeted. Now, this is where membrane fluidity plays a role, and why the membranes cannot be rigid, such as from high saturated fats, too much of cholesterol, because cell membranes are needed for SCARA5 to be able to bind oxysterols. So SCARA5, SCARA5 for short, basically binds oxysterols. There's also a three-step process, which is oxidized lipids are recognized via a cationic or hydrophobic pocket in SCARA5. Step 2, ligand binding can induce receptor clustering in the membrane, which is why the membrane has to be fluid, because if it's rigid, then the receptors don't seem to function or move around very efficiently. So if the receptors aren't functioning correctly because of rigidity, then the binding process will probably not work. And then step 3, clustering can recruit tetraspanins or adhesion molecules, altering membrane microdomains. Okay. Also seems like a calcium also plays a role because presence of calcium ions can stabilize binding in some scavenger receptors. Also, other factors and conditions are required for binding of oxysterol, such as a 7-ketocholesterol and basically a oxidative modification of the lipid. So possibly some good something to make the membrane more fluid is maybe the best or one of the best is extra virgin olive oil has a hydroxy tyrosol. Also, to avoid membrane rigidity, possibly coconut oil, because coconut oil does have a high saturated fats. Also, avoid hydrogenated vegetable oils, such as in like a peanut butter. Avoid soybean oil, which is like the number 1 ingredient in a lot of the foods at the grocery store. So if soybean oil happens to be the first ingredient on the label, then that means that there's a lot of soybean oil, or it's mostly abundant in the food. So that means that the membranes are going to be rigid and will not be able to bind the oxysterols with SCARA5. Now, something else that can really help the binding of oxysterols with SCARA5 is green tea polyphenols. Now, green tea has EGCG, and these polyphenols can upregulate SCARA5 expression at the transcriptional level, enhancing its activity via allosteric binding. Now, in order for the expression at the transcriptional level to work, the chromatin has to be opened, transcriptional factors. So once this is achieved, this leads to increased scavenging of oxidized lipids, which can reduce excess oxysterol accumulation, which is good. So also, enhanced SCARA5 has an effect on ILV formation. So enhanced SCARA5 means more oxysterol clearance, therefore reduced local membrane curvature, even fewer ILVs. So under stress, polyphenols may stabilize tetraspanins, enhancing ILV formation in some contexts. But in this context, depends on ESCRT recruitment. Okay, so a quick recap. Oxysterols bind ORPs enriched in ILV budding microdomains. ORPs plus oxysterol microdomains recruit ESCRT. Tetraspanins stabilize budding vesicles, or sounds like tetraspanins form the ILVs. Adhesion molecules such as ICAM cluster and assist curvature. SCARA5, with the help of green tea EGCG, binds oxysterols, which is the goal here. SCARA5 also affects adhesion molecule clustering, therefore affecting ILV formation, because ILVs, they need adhesion molecules. Green tea polyphenols modulates SCARA5 expression activity. Also, look at the labels on the foods. If at the grocery store, if soybean oil is the first ingredient, then that means there is a lot of that soybean oil, or the food is mostly consisting of soybean oil. Therefore, that is going to make the cell membrane rigid, which will prevent oxysterol binding. The membrane has to be fluid. A good way to make it fluid is with hydroxytyrosol from extra virgin olive oil. Some extra virgin olive oils are fake and have soybean oil, so check your olive oils. That's it for the video. Like, share, comment, subscribe, and I'll see you in the next video.